I too want to thank ICAS for the generous support of this research on fiberoptic microneedle device for therapeutic cancer vaccine delivery. My collaborator on this project is Kumran Sabai in veterinary medicine. And we had a postdoc, Charles Smith, who is biomedical engineering. Now he's transitioned off the project. He was more the engineer, helping us get the device ready to go for this application. And now Dr. Sabai has a new postdoc who just started yesterday. And to give you an overview for the significance of this project, it is that therapeutic vaccines for non-infectious settings such as cancer really do not exist. And part of the reason is that it is difficult to achieve a cellular immunity to that cancer. And what we're trying to do in this setting is target epidermal Langerhans cells, which subside in the epidermis, the upper layer of the skin, which are known to, when stimulated, promote that cellular immunity response and therefore enable cancer vaccines. Unlike the current intramuscularly injected vaccines that are common, say, for the flu. And one thing I'll tell you too is that beyond just cancer vaccines, there is broader significance is that if we can demonstrate the device works for cancer, which is a noble goal, it probably also will work for other infectious settings too, such as the flu, which recently in the news you may have heard that the elderly population has a very low chance of actually getting immunized by the flu vaccine. It's commonly thought that, oh, you go get the flu vaccine, well, now you're protected and you're safe. But in fact, only 25% of the elderly actually get immunized by the flu shot. So recently, the CDC issued statements that if you start having flu-like symptoms, make sure you get on the Tamiflu right away. So this has broader significance than just the cancer vaccine approach. So our research questions were, can we achieve a strong cellular immunity by targeting these epidermal Langerhans cells? And secondly, can we enhance the cellular immunity response using a laser vaccine adjuvant approach? And laser vaccine adjuvant is already a known effect in the literature, so that's not the novelty of our research. But what is novel, what we're trying to do is in our technical approach, we're trying to deliver the cancer vaccine or a model for the cancer vaccine in the first stages to those epidermal Langerhans cells inside the epidermis, simultaneously with the light that can act as the adjuvant, which is the promoter for the immune response. So that stimulates those dendritic cells to uptake the vaccine and put out the right antibodies to protect against that. And so our approach is with this fiberoptic microneedle device, which has been mentioned in a few other talks, which are hollow glass needles that allow light delivery through the annulus of the material and then drug delivery through the bore. And we're going to target them into desirable locations in the skin, the epidermis primarily, and deliver the vaccine plus the light. So we've done that. That's our achievements. We've developed and prototyped multiple different types of FMDs. This is an example where there's a standard syringe needle as well as a standard light-only delivering fiber optic, and they come together under this sheath area and get combined in this novel area. So this is what we've patented with numerous provisional as well as U.S. utility patents now, is the manufacturing and the application of this co-delivery fiber optic splice. And so in the end, you get light and drug out of a low gauge, thin needle that you can insert into tissue. And this demonstrates the light delivery, which we can also modify the surface of the fiber optic to tailor where you get the light delivery out of the fiber. So we've applied this in ex vivo rodent studies, which is mice skin, and we've used a model vaccine, which is enhanced green fluorescence protein, and delivered this into that ex vivo skin and then counter-labeled the EGFP with another immunostain, and that shows that it works because of the epidermis which is this dark brown immunostained color. So the approach we've demonstrated is now feasible. So our future directions in the second year of funding is we'll start with live mouse studies with FMD. So we're working on our ICOC approval, and I'll get to that on the next slide, the live mouse studies. And secondly, this whole idea in brainstorming with Kumaran, we've developed some other ideas that we're going to work on in parallel to this fiber optic microneedle technology. And one is that we're very excited about applying optical clearing agents, which I have a strong background. I myself have a strong background in using dimethyl sulfoxide and glycerol, these hyperosmotic agents, to change the optical properties of tissue. And that's shown here in these pictures, showing a ruler, and you put a piece of skin above the ruler, and it's opaque, so you don't see the ruler anymore. But if you apply these optical clearing agents, you can, with time, dehydrate the tissue, basically, index match the tissue, and make the skin transparent. And it's a reversible process. Dr. Sabai has applied those same agents just by coincidence in his own line of research in the past years, showing he can deliver vaccines with them because DMSO is a penetration enhancer. So another technology that's going to come out of this is combine optical clearing agents and vaccine transcutaneous delivery methods. So we're going to do that research in parallel. So we're going to write grants. One is an R21, which we nearly submitted this last cycle, but we felt we would hold off to get a little bit more preliminary data, so we'll submit that in June. And an R01, hopefully based on our optical clearing results in the fall cycle. Just real quickly, do I have a couple minutes to go through a couple of chart things?
You sure do.
Okay, because I thought I had a few. I won't go over the highlights because I think real quickly, what we're going to do in moving forward is with the live animal studies, we'll look at this laser vaccine adjuvant effect on the CD8 plus T cell immune response to first a model ovalbumin vaccine, and secondly to Dr. Sabai's novel peptide-based cancer vaccines in a melanoma model. So that's our future work. We have some publications in preparation, and I wanted to point out that we've gotten preliminary provisional patent on this application of using microneedle for therapeutic cancer vaccines. We'll need to convert that in this August, or else we'll lose our IP rights on this. So I think that's something we're going to have to figure out how we're going to get that money, because that'll probably be about $10,000. So I'm working on converting other applications in the works. So hopefully this can get wrapped up by a licensee. So that's what I wanted to point out, is that I really think the future funding for this project is going to come through the industry side. And so I've engaged numerous potential investors from UNC as well as BT alumni who found interest in this, and we're in the talking stage right now. And it's actually moving along faster than I thought. Also, we'll be able to go to this TechConnect Innovation Summit in Washington, D.C. to pitch the technology and hopefully get more outside investment. And that's all I have. Thank you.
All right, so we have Kumar, and he's going to give us two back-to-back talks here. So you can just take us up to the break. All right.
All right. Can you hear me at the back? Traditionally known as a mongler. So
What was that?
Please. All right. So I have to thank TFS for supporting me on several projects, seed funding, and let me start with the chemocompatibility of drug-related nanomaterials. This is a collaborative project, interdisciplinary, with Dr. Riffa, who is our nanoparticle person. She has been working with nanoparticles for several years, and she has come up with fantastic nanoparticles, polymeric nanoplexes, magnetite nanoparticles, to name a few. And we have been using those nanoparticles to study their behavior in terms of how they interact with blood components, and as well as whether we can deliver drugs through them, and whether the drugs will actually be efficacious when we deliver through nanoparticles. So the other member of the team is Dr. Sri Ranganathan, and we have been collaborating for the past several years. So in this particular project, we have been trying to study whether these nanoparticles are actually toxic or become toxic when you deliver intravenously. So what we wanted to study is study in vitro methods to characterize hemocompatibility. Hemo means blood, compatibility in blood when you deliver systemically in humans or animals. And then also take it to the next level to study the hemocompatibility in the animal model. So what we did is, the first phase of the project, we used co-block polymers, which are polymeric nanoclusters, which have been